Welcome to the BOSC function cozy joint session. I'm Iddo Friedberg, and I'll be moderating this. And this idea is sort of the brainchild of Nomi Harris and myself. We were talking for a while about doing some joint sessions. The function cozy, which I'll talk about in a minute, is really relies a lot on, like all of bioinformatics really, on many different open source projects like the ones that are being here. And we actually rely on a community to produce quite a bit of our software. And I was thinking this is a great way to get both communities engaged and talking with each other. So I'll talk a little bit about what we do in the function cozy. We do a lot of things. We basically are looking into doing protein function prediction using computational methods. And one of the projects that we are running, our main project, is the critical assessment of function annotation, which is, like many other computational challenges, basically a kind of a competition between different groups and how well you can predict protein function. And as we know, there are many computational ways to predict protein function. You can predict it from the sequence by homology search. You can predict it by the genomic neighborhood, by synteny, function, phylogenetics, structure. And you can feed all sorts of attributes into your machine learning method or something even simpler and get some really interesting results. And the critical assessment of function annotation, or CAFA, tries to sort of standardize all of this and give scores and find out which methods are doing well and how they can improve. So it's a community challenge, pretty much like the Kaggle or the other that you are familiar with. The way that we assess these methods is pretty simple. Let's say the true function of this protein is DNA binding. On the gene ontology, this gets propagated into more general terms, nucleic acid binding and binding. Whereas the predicted function, you might have predicted that it's ribosomal RNA binding. So you were wrong, but not really wrong. You predicted indeed that it's nucleic acid binding and the function is binding, but the wrong nucleic acid. And then you can start playing around with how many true positives you have in your prediction, how many false positives, and create some sort of precision recall curve. And these are the results that we have from one of the CAFA. Then you propagate that onto many different proteins and many different organisms, and you can start ranking the methods. I actually redacted the names of the methods here, but you can see how well you're doing in relation to BLAST, and you can see a lot of methods, at least the top methods, are doing better. These are precision recall curves. The ones that go here are better. So here we have a method that really is doing much better than all others. And those are the kind of things that we do. This requires a lot of data wrangling, a lot of good code to assess the methods using different metrics. We need to managing participants by itself is a big task because we have so many of them. We had 140 methods participate in the last CAFA, no, 160 methods, sorry, participate in the last CAFA out of many different groups. So just the management of that is a huge effort. There's a lot to do here in terms of code, and you can learn about that in our website, biofunctionprediction.org. And you're welcome to contact me or joining our mailing list. We also have a Slack, which I did not put here, but I will at some point in the chat. Sorry about that. And there's a lot of interaction that should go on here. So I will stop this little introduction, and I hope that we can discuss more about how this community in BOSC, which I know very well, I've been attending BOSC since 2000, since ISMB San Diego, will be able to help and contribute to CAFA. And seriously, all our software is, of course, open source licensed, And we are looking forward to working with this, hopefully working more with this community. I'd just like to thank the people involved in CAFA, Predrag Divoyats, Casey Green, and Sean Mooney, Sage Biosciences, who are hosting a big part of our bioinfrastructure, and Lara Mangravidi will be talking later about that, and all the CAFA participants. I'd like to thank from the Function Cozy, my co-organizers, Mark Wass and Kim Reynolds, and BOSC. I'd like to thank Nomi Harris for helping, for working with me to make this happen. So that's it. And now I'd like to introduce Monique Zahn from Swiss Institute of Bioinformatics, who will give a talk.
Hi, I think we're showing the video actually.
Okay, who will be showing a video.
Hello, everyone. My name is Monique Zahn, and I work at the Swiss Institute of Bioinformatics. Today, I would like to convince some of you to complete the functional human proteome together with the NextProt team. To begin with, let's take a look at what we know about the human proteome. So NextProt is the SIB knowledge base on human proteins. It provides data taken from a number of different sources and tools to analyze these data, thus providing a platform to explore the universe of human proteins. Now, the data is all available through the CC BY 4.0 license, and most of the code is also on GitHub. So what do we know about the human proteome? Well, let's consider the Human Proteomics Organization, which launched the Human Proteome Project 10 years ago or 11 years ago. And their aim was to get evidence from mass spectrometry for the presence of as many as possible of the 20,000 protein coding genes that humans have. And to do so, we will consider the protein existence value that is in UniProt, where when there is experimental evidence at the protein level, it's PE1. When it's evidence at the transcript level, it's PE2. The value is PE3 when there's evidence coming from a species other than human. PE4 when the evidence comes from a gene model, and PE5 means it's rather uncertain whether there is protein exists. And at NextProt, we use exactly the same PE value scale as UniProt, and we also apply the same use to assign these values. Now, since we have data from a number of additional resources, we can upgrade the protein existence values of entries which are PE2, 3, or 4 to PE1 with mass spectrometry data taken from Peptide Atlas, Massive, and NextProt, and to PE2 with RNA-seq data from Human Protein Atlas and BG. Last year, the HPP project reached a major milestone in that over 90% of the predicted human proteins have been detected. This came out as a Nature Communications paper, and NextProt being the reference database for this project, you can see here for the current release that just over 90% of the entries currently have evidence which is PE1, so at the protein level. Let's now turn to the functional human proteome. What do we know about the function of these human proteins? In NextProt, we have this information in the protein function view. So I've shown here the example for MSH6, which is a DNA mismatch repair protein. And we provide information about function at 3 different levels. At the molecular level, for example, if it's an enzyme, what biological processes it's involved in, and what pathways it is in. Now, to look at how many proteins do not have a function associated, we have this Sparkle query, query 22, in which we exclude proteins which are PE5, and we also exclude those which have annotations to very general gene ontology terms, i.e. protein binding. And we see that there are 1,669 such entries in NextProt. So let's try and predict the function for some of these proteins. For the last 2 years, we've been organizing at the University of Geneva a course which is called Fonctionnaton. And in this course, the undergraduate students get training about databases, bioinformatic tools, and they are then assigned one of these proteins, which doesn't have a function. You see here, it just has protein binding that's known about it. And they're divided into groups of 2 to 4 students, assigned a tutor, given the protein, and then they go through a manual data mining workflow, which consists of what a biocurator would do. So they extract information from the literature, so they do searches in PubMed, they compile data from databases, and they run bioinformatics tools on the sequences and so on. And they take all this information and build a protein identity card, consolidating the information that they have on the protein. And this enables them to formulate the hypothesis as to what the function of the protein could be. And they also propose an experiments to test these hypotheses. And the tutors then standardize these hypotheses using gene ontology and evidence code terms. So if we take the example I've shown here and take a quick look at the ID card, we see that we have all the names for the proteins. This is, of course, important to know what you're going to be searching for. Because in essence, what the students are doing, they're acting as profilers on these unknown proteins, trying to gather information and trying to determine what the protein does. So they will look at the conservation across species, they will look at domains and PTMs, they will look at where in the cell the protein is located, what other proteins it interacts with, where it is expressed, and also what phenotypes and diseases are associated with this protein. And they will come to a conclusion and propose a hypothesis. So in this case, the students proposed that this entry had ribonuclease activity. And this is the Go molecular function term, ribonuclease activity, for this entry. Now, these predictions of the function of the proteins are now displayed in NextProt, as you see here. So it's displayed in a community function prediction page for the entry. And we see here the ribonuclease activity term we saw for the entry with the orchids of the tutors, and a number of different evidences representing the different type of data supporting this function. So what's next? Well, I started off saying I would try and convince you to join us in this endeavor to find the function for the remaining human proteins. So if you wish to do so, please use the contact option in NextProt, send us your ORCID, tell us which entry you've selected from the query, tell us what you think the protein does and why. If you can't express this as terms, we'll be happy to do so. And if the data comes from a publication, please provide the PubMed ID. And we will post your function prediction. So on behalf of the NextProt team, thank you very much. And I'll be happy to take questions. Thank you.
Okay, thank you. Thank you, Monique. I understand that my slides weren't showing during my talk, so I think that's actually a blessing in disguise. Hopefully everything made sense or not. We have a couple of questions. So the first one from Chenxing Zhang. how the general Go terms in Nextprot are defined, i.e. what is the mathematical criteria to consider a term being too general or specific enough, which consider which proteins are functionally annotated. Sorry, who comes up with these criteria?
Okay, so in fact, these criteria came up through interaction with the HUPO community, and they were selected on the basis of the knowledge from the annotators that annotate the Go, and knowing that something like protein binding usually doesn't give you very much information, especially when you don't know which protein it's binding to. So that won't help you predict a function if you don't know what it's binding other than it's a protein.
Okay.
So there's no real mathematical criterion. It's more a annotator knowledge criterion.
Okay, thanks. Well, we can continue this offline. Then the next question is from Itamar. Can you please clarify if Nextprot is replacing UniProt? I'm not sure I understood the distinction.
Oh, so no, Nextprot is not replacing UniProt. UniProt covers all different species. Nextprot is uniquely focusing on proteins. And although we take some of the information from UniProt, we do take quite a bit of additional annotations from many other sources. So there are two separate projects. And if you want to remember, the distinction is think about the question, what comes after UniProt, what's next, and it's Nextprot.
Yeah, UniProt is also focused on proteins, so the difference.
Yes.
Yeah.
Yes.
Okay, so Nextprot is focused on proteins.
On human proteins.
On human proteins. Okay, all right.
Exclusively human.
Exclusively
yes.
human. Okay, all right. That's the
yes.
part I missed. Thanks. All right. Are there any other questions? We'll wait a couple of minutes to see if people are typing in.
Otherwise, I'm happy to answer on the Slack as well.
Yep. The Slack and BOSC is extremely active, I might say. It's a really nice community. Okay. Oh, there's a question from Peter. Is there a URL for this community annotation project with Nextprot?
No, there's no URL for this community project.
Okay.
Maybe we should think about putting one.